am a neuroscientist. I have a PhD, and I'm one of those people who does not insist on being called doctor. And one major reason for that is I do, in fact, work in the clinic in the hospital. And when I go in to do a study, and I go into patient room, very commonly I will walk in wearing the white doctor coat for my first approach to a patient. Hi, I'm Dr. Hampson. I'm the lead principal investigator on the study that you agreed to participate in. Let me tell you a little bit about the study, what we're going to do. We're going to come in here. My team's going to come in a few minutes. You will not see me in this white coat again, at which point I take it off and I'm wearing a Hawaiian shirt. I am a doctor. I am not their doctor. And so as a matter of fact, lots of folks feel obligated to call me Dr. Hampson. It's fine, I won't stop you, but you don't need to. That's the whole point. Because folks know me as Rob or Speaker, and folks who have known my pen name for many, many, many years past will also know me as Ted Roberts. And there were many times when there was confusion as to which of the names was my actual name and which one was the pen name. And for a while, people thought the pen name was the real name and the real name was the pen name, and that was fun.
Then there's this speaker.
Then there's this speaker fellow. Because back in the days of Usenet, because I am that old, I had a signature line in the Usenet groups. Speaker to lab animals, mind reader to Rattus norvegicus, Macaca mulata, and the murine at the other end of the bar, meaning rats, monkeys, mice. And at the time, I had just developed some novel recording and analysis techniques that allowed us to read patterns from a part of the brain that was involved in memory that actually helped us predict what the rat was going to do in its behavioral tasks, because we could see a representation of the memory that showed us whether or not the rat was going to correctly retain and recall that memory at a later point when it had to do so in order to make a response and get a nice little reward. Now, rats work for water, monkeys work for juice, humans work for beer, or bourbon, or coffee. So if you will excuse me a moment, one of the things I am going to do right now is I'm going to pour myself a cup of this coffee, because I put it in the thermos so that I can get to the panel, because we're at con, which means we don't get enough sleep. And when Aaron asked me would I like to do a panel today, and I could do a free form, I could do pretty much anything I wanted to do, I said, okay. I'll talk to you all a little bit about state of the art, what we're doing, what I'm doing, what neuroscience is doing these days. Talk a little bit for those of you who here is a writer or wants to be a writer. Okay, so we're going to talk a little bit about the brain tropes I would like to encourage you not to use. And then open it up to a Q&A. Because, I've done this Ask a Scientist, Ask a Neuroscientist, come in, free-form Q&A with Dr. Rob is great, and I love doing it. A lot of what I do is public outreach and education in the sciences. I'm training students to be able to do that, because scientists need to be their own best advocates. We've just lived through 3 years of nobody really knowing what the real science was behind anything, and everybody claiming they had it, and also claiming the science was settled. Science is never settled. If science were settled, it wouldn't be science, because the whole point of science is asking questions. Which is my segue into the conversation itself here, which is where are we now with the state of the art in brain science? My new book deals a lot with that, because 40 years ago, I was getting ready to go, 45 years ago, I was getting ready to go into graduate school for the very first time. We've already talked about the fact that 50 years ago was first airing the first episode of The $6 Million Man. I loved it. I wanted to do this. This is what I wanted to do. For my first thoughts were I wanted to be Rudy Wells. I wanted to work on the bionics, be the neurosurgeon. I was actually told by a good friend, you know, you really probably don't want to be a neurosurgeon because their patients are in pain and they never get any better. I actually discovered, pun fully intended, that I didn't have the patience to be an MD. I love research. I love investigation because if you do it right, it's always something different. And so I went into research and I wanted to go into bionics. And the field didn't exist. Prosthetics research in 1979 meant trying to find the best alloy that would not react with the saltwater environment, the electrified saltwater environment of the human body. And I discovered that the hard way because I went to an engineering college where I thought they were doing this and found out that the professor who was doing prosthetics research was a material scientist. And I was a biologist. I wanted to do biology. In 2016 through 2020, I was heavily involved on a DARPA project to study memory with the intent of fixing broken memory. Can we identify when memory doesn't work right electrically by recording from the appropriate parts of the brain and supply a signal? Is it a drug? Is it an electrical signal? Is it something else we could do? Could we inject stem cells and regrow the area? What could we do to make memory work properly when it hasn't been working? DARPA funded this. They also funded a group called Subnets, which was exploring the science behind deep brain stimulation. People who have Parkinson's disease and have severe tremor, when nothing else works, we can put an electrode into what's called the subthalamic nucleus. And we stimulate it. And what we're doing is signals out of the subthalamic nucleus are abnormally synchronized. It's not true. Trope number one. It is not true that we only use 10% of our brain. We use all of the brain all the time. We just don't pay attention to it. But it's asynchronous. This part of the brain is not running the same as this part of the brain, is not running the same as that part of the brain. They each have their own rhythms, they have their own specific activity, and there are many different factors that go into what the brain activity is. Synchronous activity is bad. Highly synchronous activity is called a seizure. And we don't want synchronous activity. And one of the things is, even though it's not so broadly spread as to cause a seizure, tremor in Parkinson's disease is caused because there's synchronous firing of neurons so that the real signal can't get through. But we can break that signal. We stick an electrode in there and we stimulate it at anywhere from 100 to 200 hertz, and we can break that synchrony, and the muscle movement signals can get through. Deep brain stimulation. Well, hitting that target can be hard for a neurosurgeon. We've gotten really, really good at it, but in the early days, there would be electrode placements that were a little bit off target. And they had interesting things. Persons who were having problems with anxiety and depression said, hey, I don't have anxiety and depression anymore. Individuals with OCD may find a lessening of their OCD tendencies. There are a number of different side implications for deep brain stimulation. DARPA was working on that, and in a program called Subnets. What are the other things we can do with the stimulation? And then we had the entire question of prosthetics, advanced prosthetics. Could we provide a person with an artificial arm and hand that was more than a hook? And a program that ran for 12 years called Revolutionizing Prosthetics. It actually had 3 iterations in DARPA, RP1, RP2, RP3. In between 2016 and 2020, when I was going to most of these meetings, they were in RP3. RP3 had created 3 products. One was an advanced arm that came from Johns Hopkins University Advanced Physics Lab. And I have shared the pictures in many talks past, and people who look this up have probably seen the picture of Johnny Matheny with a titanium rod coming out of the stump of his upper arm, a magnetic bearing elbow, and a fully articulated hand. And that fully articulated hand is not the most flexible, it doesn't have all the functions that we put into it, but for its time it was the most advanced one. I've met Johnny, very pleasant fellow, has some amusing stories to tell, one of which is the fact that a second iteration of the arm, he insisted that it had to have a wedding ring built in on the ring finger. And he actually has learned to play piano with it. He did not play piano before. He has learned to use it to play piano. So it's very flexible. The second development out of the program was what we call the Luke arm. And the Luke arm was put together by a company called DECA. And the name DECA comes from the first 2 letters of the first and last name of the person who founded the company, Dean Kamen. Dean Kamen is probably best known as the guy who built the Segway. That little platform with the 2 wheels and the stem that you see mall cops running around on and things like that. So Dean came and put this together. They built an arm that can be prescribed and paid for by insurance and Medicare, and that amputees can wear, and particularly military amputees. And this one it's covered by a translucent cover that can then be painted and tinted to look more skin-like. To me, it tends to look a little squashed, a little flat, but it's fully articulated. Many more movements of the wrist and the lower joints of the fingers than the APL arm that Johnny wears. And some of them actually have tactile feedback, because you can put an electrode here in the motor area and drive the hand muscles. But that doesn't get a sense of touch back to the brain. Getting a sense of touch back requires another electrode in an area that processes sensory information. And they worked on that. The third product to come out of RP3 was the fully articulated robot hand, completely controlled by electrodes on the patient's brain of quadriplegic and even locked-in patients. This is a device that can be mounted to a wheelchair, or it can be substituted with wheelchair controls. And the electrodes are on the motor area and they're on the sensory area to provide force feedback and everything. And there are pictures. There's a 60 minutes video of this where you see the woman feeding herself a candy bar or reaching out and manipulating things on a board. And it's a fully robot arm. It's not wearable, but it can be configured to be transportable. And as with many, many things, the computer systems used to drive all this keep getting smaller and smaller. I made a decision to go into physiology, and at the time, I started graduate school in '79, somewhat disappointed by not having a bionics program. In '82, I started a PhD in physiology. And around that time at Wright State University were a set of experiments where a woman who was paralyzed from the waist down was given the ability to walk. Electrodes in the muscles of the legs would stimulate and contract the muscles and allow her to do a walking gait. There were also electrodes in the spinal cord that helped to provide signals so that she could maintain her balance. Originally, the computer required to support this would stretch from one end of this stage to the next. By the time she graduated from college, they were able to do it with a computer about half the size of this desk. The computer systems used for the robotic arm and part of RP3 probably about 10 of these books needed to control it. We have lots of other instruments and amplifiers and connections and monitors and everything else that we add to it to make something, that you would have to put on this desk. But the technology keeps getting better. Computers always interested me, which is why I ended up in this field. But between 2016 and 2020, we stopped having the in-person meetings then. But our project to study memory met along with all of these other projects. And in 2019, I looked around the room and realized the science of bionics was here. And these people in the room created it. And that was pretty cool. And that's one of the reasons why I wanted to give back in the form of this book. Those who have read it, those who have reviewed it, have commented on the fact that we see an awful lot of the rehab. I did that on purpose. I wanted the reader to understand the sheer courage that these volunteers have gone through in order to bring us this technology to a level that will eventually become commonplace, although it is not now. Adoption of these prescribed limbs is low. The costs and the reprogramming and the tuning and everything is intrusive, because you have to keep going back to the doctor. You have to keep going through physical therapy and rehab as if it were an injured arm you were trying to rehab. But it's there, and it's possible. And that's the state of where we are right now. There's a hand that's programmed by your iPhone. So you reach out with one hand, punch a button on the iPhone, and actually now they're becoming routines, so they use pressure sensors in the hand. Reach out, grab the cup, and when the pressure sensor triggers, it grasps the cup, and then you come back and drink your coffee. And by the way, there's the proximity sensor so that it tilts when you get here. This is for not an arm, but just a hand, because that's another thing that's necessary. During one of the focus group sessions before RP3 and these other programs became a reality, the focus group said, what do you want as people who would be the users? And most of the young people said, I don't want electrodes in my brain. Okay, that's fine. We're working on that technology. We have a number of different fascinating techniques that range from trickling DC current from one side of the brain to the other via scalp electrodes, but the current goes straight through. That can help you go to sleep if you have insomnia. We've got the ability to stimulate deep areas of the brain without having to stick an electrode in by using focused ultrasound. And ultrasound goes in and it spreads, right? Anything sound starts at a point source and it spreads. But what if you take 4,000 point sources and you time them so that instead of spreading, the signal spreads, but everywhere those waveforms cross, they either reinforce or they cancel, and you can focus that down. And focused ultrasound gives us a means to do surgery. It's actually been commonly employed in Parkinson's disease surgery. Again, if you kill off or damage the cells that have this abnormal synchrony so that the signal can get through, then there is a possibility for some therapeutic improvement. But let's not use that much energy. Let's just use it to tickle the neurons and get them to be more active. There's a non-invasive way that we're working on and people are working on to develop it. Well, how do we read this stuff? If we read the EEG, that's coming here from the scalp, and frankly, it's difficult to isolate all the different pieces of activity from deep in the brain. And all your information doesn't come from here. All your information is coming, it's all through here. It's the base of the brain. It's the brainstem. It's the auditory areas. It's the what we call the temporal cortex, the prefrontal. All the deep areas are all part of processing the brain. If we're going to record and we're going to stimulate, we have to access those deep areas. And right now we do it with little metal electrodes, fine wires that are no bigger than a hair. But if we can do it without even having to open up the head, that would be even better. So there are now techniques to use what we call optically pumped magnetometers. Optically pumped magnetometers, or OPMs, are little smaller than your fingernail that can be placed next to the scalp and pick up electrical signals, magneto-electrical signals. And if you can put enough of them on the scalp and you have a quiet enough environment, you can start recording, and having enough of them in a three-dimensional arrangement around the head allows you to triangulate and figure out where signals come from. So that's another means we have now of doing the OPM, doing the recording from inside the brain. There's optical techniques. We already know that we can take the MRI machine and we can look for oxygen signals, oxygenated blood. And if we look for areas where there is a differential between the oxygenated and non-oxygenated blood, well, the oxygen had to go somewhere, which means that those brain tissues are active. They're functioning, they're doing something because they've gobbled up the oxygen out of the blood. So we have another technique for saying what parts of the brain are active using the MRI machines. We can put a person in a magnetoencephalograph, very large device and system that is excellent for recording. It requires a room that is absolutely magnetically isolated. It's got to be shielded, it's got to be filtered, we've got to build counter fields into it that will cancel anything outside, and the devices have to be bathed in liquid helium. Which is a small problem. We have a helium shortage in the world right now. And it absolutely amazes me that there are party stores where you can still buy helium-filled balloons, because there was word about a year ago that would absolutely stop. The only reason why Macy's company is allowed to use helium for their balloons for the Thanksgiving Day parade is because when the parade is over, they suck all the helium back out and they run it through a recycler and separate the helium from the air components, and so they reuse about 97% of the helium. But helium is much more important because liquid helium is how we cool the magnets in our MRI machines. It's how we cool the superconducting quantum interference devices that make up our new quantum computers and the magnetometry, the magnetoencephalographs. So there are a number of different things, but as always, the scientists are working on making it smaller, more portable, and that's part of our state of the art. So on to tropes. First thing, we talk about the lobes of the brain. I always tell people to put their hand up and make a fist, just like this. And when I was talking about this on Writer Dojo, and we were recording audio only, and Larry and Steve and Jack all put their fist up and did it exactly the same way. So, front of the brain, back of the brain. The front, the frontal cortex, and the prefrontal cortex, right just above it, is where a lot of what we consider thought takes place. Executive function, cognition, decision making. The very back, here, at where the wrist would be, that's the occipital lobe, and it's in the back, and that's where most of our visual processing is, as well as some of the parts of the brain that are involved in putting together vision and from small pieces into big pieces so that we can identify things. Now, some of you know about the cerebellum, which is essential for coordinating our movement. Cerebellum is kind of tucked up in here under the wrist bone. In front of the knuckles, we have the frontal lobe, and a lot of our motor control takes place in this area right along the front side of the knuckles. Back side of the knuckles we call is parietal lobe, and just immediately on the back side of the knuckles we have the sensory area, where there's a representation for every part of the body, and most of our touch and position and movement sensations are representative. Between this occipital and parietal is a number of what we call association cortices, which is where we put together vision and touch, vision and sound, sound and touch, to start putting together the full picture of what's in our environment. This thumb down here, sticking down here, pretty much like a catcher's mitt, is the temporal lobe. And the temporal lobe is very important in memory. There's a large structure called the hippocampus. The hippocampus is one of the major areas involved in memory. And from back in the 1950s, when a young man by the name of Henry Molaison had had epilepsy surgery, and the doctor went in and scraped away the inside surface of his temporal lobe on both sides, discovered that he could no longer make new memory. We call this anterograde amnesia. He's pushing forward, as opposed to retrograde amnesia, which is looking backward. Anterograde amnesia means not able to create new memory. He could remember something for about 10 minutes and then he would forget it. He couldn't turn it into long-term memory. He could remember his past, but he couldn't make new memory. So we know the temporal lobe is really heavily involved in memory. It's also involved in emotion, in particular, the emotional content of memory. It's involved in the history, understanding what happened before and how that affects what happens now. So those are the major parts of the brain. Trope number 2, stay away from it. Total amnesia. Soap opera amnesia, telenovela if you like. So it's in the soap opera, our tragic heroine gets conked on the top of the head, has total amnesia, does not remember anything about her past. So she gets taken in by friendly strangers who are going to help her. Maybe they say, oh, we'll help you remember who you are, recover your memories and everything. She falls in love. She discovers that she has this wonderful life, and it goes on for 7 to 10 years, and then bonk, she gets hit on the top of the head again, and all of her memory comes back, and she realizes that she had a husband and kids. And it's tragic, because now she's torn between 2 lovers, and it doesn't work that way. Okay, so number 1, bonk on the top of the head. That's not going to be what causes amnesia. Impact or blast injury on the side of the head. Something that's going to affect the hippocampus, something that's going to affect the parietal and association cortices along here. Blast injuries are particularly bad because the pressure wave hits and you go this way while your brain goes that way, and then you go that way while the brain goes that way. And so you have 2 bruises on the inside because the brain is sitting in a bath of liquid. We call that coup contre coup, right? Because
The paramedic knows that.
I could tell. So, and the injuries of that sort will cause amnesia. But total amnesia is rare. We don't see total amnesia so much as we see partial amnesias. So what are partial amnesias? Now, here's the advice to writers part. It would be so much more interesting if after the head injury, your patient had an agnosia. I don't know. Agnosia, I don't know. There are pieces of information they cannot access. It might be names, in which case it's proper noun aphasia. They can't speak. They can't remember, this is a thermos, this is a book, this is a microphone, that is camera. That's a picture thingy that moves, and you can look at it later. You know, that's aphasia. Where do we see that? We see that with stroke. We see that with stroke because there is a localized injury to the brain and not a large one. Because unless this egg is cracked open, the injury is much more likely to be local instead of the entire brain every time. So an agnosia, or an aphasia, or a neglect. Oh, the neglects are fascinating. Neglects have a tendency to take a large chunk, affect a large chunk of visual and association cortex. I can see what's over there, but if you ask me what it is, I couldn't tell you. My visual system's intact. If there's a moving object, I could probably track it, but not be able to tell you what it was. That is a neglect. There are conditions called hemianopias, where one half of the visual system is lost. When we see with our eyes, any given eye, you notice you can see the whole field. I can't see right back there, but I can see from there to there with one eye. You're actually using both halves of the brain. From here to here, on this side of the brain. From here to here, that side of the brain. Both eyes. So for this eye, looking over here, comes back to the side of the brain. This eye, looking over here, same thing. Your two visual fields are set up so the two eyes split, and whatever is to the right of the nose goes to one half of the brain, to the far opposite side of the brain. Whatever is to the left of the nose goes to the opposite side of the brain. If you have damage to the eye, you still have the full field. If you damage the brain, you lose half. Hemianopia. Hemi, half, and opia, no optic, no vision. If you want to have a head injury in a patient, if you want validity, look up coup contra coup injury. Don't give them total amnesia, give them a partial amnesia. Give them an aphasia. As I said, we see aphasias a lot with stroke, and we also see it with traumatic brain injury and other things, but strokes are fairly common. And with aphasia, it's the most severe is an inability to speak. The intermediates are an inability to speak certain types of words, nouns, names. I worked with a person who had a proper noun aphasia. He would grope for the name of something, and his compensation mechanism was me. I'd fill it in for him. I'd tell him who the person was he was talking to, or as the person he was talking to walked away, I'd say, yeah, that was George. So George who? George was your graduate student 10 years ago. You know, that's the sort of thing we see with memory deficits as they start to build in. And the neat thing about aphasias is you can bypass it. One of the fascinating things neuroscientists have found is that when you have an inability to speak, you still have the ability to sing. And I know of several therapy programs where they teach stroke patients who are having speech difficulty to hum and sing song and sing what it is they're trying to say, and it comes out. Because what you actually have is you have a speech center here. People who've heard of Broca's area. This is the area that helps you program the muscles to make the speech happen. There's another area back here just above and slightly behind the ear called Wernicke's area. Wernicke's area is the area that's involved in language. So forming the words in your head is a function of Wernicke's. Wernicke's is also involved in understanding what we hear and read. Linking the two deep is something called the arcuate fasciculus. And lots of times in stroke and head injury, while the surface connections which would connect these two areas may be damaged, or the areas themselves may be damaged, the arcuate fasciculus is intact. The arcuate fasciculus is what we use to generate music. And it helps us to sing, which is one of the reasons why, if you want something really, really fascinating for a character, they've had a head injury, they have an aphasia, and they've learned to sing in order to communicate. Meltilus, with his incredible stutter, hums. He gets started by humming, and then he can sing just fine. And I've got a friend who gets severe migraine and cluster headaches, and he gets aphasia. And I told his wife about the possibility, try to sing. Well, he didn't apply it directly. What he did is lower his voice into the singing range. And as he's talking now with his voice in his singing range, he is able to communicate when the aphasia hit too hard. Trope number 3 is that our brains grow until we're 5 and then they stop. Trope 3.1 is that our brains are fully mature by the time we're teenagers. The truth of the matter is that the brain, certain areas of the brain can continue to create new cells and new connections all the time. It's specialty areas. The new connections happens all the time. That's how we learn. The new cells happen in certain brain areas in which there's a lot of turnover, and which it's very important for these new connections to take place, one of which is the hippocampus, the area that I study in memory. But I have some sad news for the people who crave a certain recreational leaf. I've done a lot of study in the field, and there's good, bad, and ugly, as with anything, as with any pharmaceutical. I don't believe in medical marijuana. I do believe in medicinal cannabinoids. In other words, control the dose, control the route of administration, make sure you've got the right compound, the right constituents for the indication. Because in terms of combating, correcting the spasticity that comes with multiple sclerosis, or working with glaucoma, and very, very important, combating the loss of appetite we see in cancer chemotherapy patients. The effects of the cannabinoids are quite beneficial when controlled, when dosed appropriately. We are finding that some of the non-psychoactive ingredients may be very important for seizure control and convulsions, and for some protections against overheat and underheat. A person who gets severe heat stroke can convulse, but if we can protect them in some way, then that would be quite beneficial. And there are many, many benefits. But people like to claim that it's absolutely harmless. Well, it's not. It has an effect on the developing brain. So then the point then is, okay, then you keep it out of the hands of kids. Well, your brain doesn't fully mature, particularly in the parts that are involved in assessing risk, making decisions, until at least age 25. In particular, what's happening is the neurons are developing an insulating sheath called myelin. And frankly, if I had to get a signal from here all the way down to my toes, the electrochemical reactions that make up neural signaling aren't fast enough to keep me from stubbing my toes, stepping on the Lego, or stumbling over the hot coals in the campfire. One of the ways we make up for that is multiple pathways, some of which have only 2 connections before you get down to the muscles of the foot. Some have multiple connections, so signals come at different speeds. The other thing is we insulate the neurons, because anyone who knows electrical capacity, you lose signal the longer the wire. But if you can insulate the wire, you can actually preserve the signal. You can preserve the strength. The other aspect of it is this is all electrochemical. So it doesn't just an electron moving from one end to the other. It's actually electrons running in little itty bitty loops in increasingly small segments of the neural membrane. But if you insulate those, then they jump from node to node to node much, much, much faster. That insulation coating, particularly in the frontal parts of the brain, is not complete until sometime around age 25. So it's neither the case that the brain stops growing at 5, nor at 18, nor that we're fully mature from age 13 onward. Our brains are always developing. Trope number 4, once you lose it, it's not coming back. But the brain is incredibly flexible, it's incredibly plastic, and it can recover. And mostly what it does to recover is it says, hey you're only doing one job, now you're doing 2. Because lots of different brain areas are serving multiple functions, there's a lot of redundancy. So we can recruit an adjacent compatible brain area to take on some of these functions. The other thing that is trope 4.5, because it's not quite trope 5, but it's not that close to the first one, is that since our brains are divided into 2 halves, the left side controls the right, the right side controls the left, that if we lose function over here, we'll never get this back. And the truth of the matter is we can actually get some of that plasticity. The younger the better. There are children born with a severe hydrocephalus in which they really don't have a developed one half of the brain. It's not well developed, and yet they still can function perfectly well, controlling the whole body with one half of the brain, because it all takes place in that 25 years of development, and then everything that comes past that. Trope 5, and then I'll go on to some questions, is the left brain is precise, mathematical, analytical, and the right brain is creative. To quote Jason Cortova, this is some bullshit, because it doesn't work that way. Yes, most of that is actually based on the fact that language and speech appear to be lateralized to the left side. And most of the people, and it also comes about with people who are right-handed, obviously controlling the left, gravitating toward the more analytical, the more numerical computational fields, and people who are left-handed gravitating toward artistic fields. That's a matter of preference. And statistically, and actually it's anecdote, because statistically it doesn't really add up. And when we look at the left sides and the right sides, we don't see a whole lot of difference. For a lot of this to go into a lot of the deeper dive in some of the things I've talked about with these tropes and the principles, I would invite you to go to Baen.com and look at the free nonfiction starting from the year 2011. I've been writing articles, both with Ted Roberts and with Rob Hampson, for a one-stop shop to find them. At my website, rehampson.com, I have a list and links to as many of those articles as possible. And I invite you to go and do a deeper dive. I had actually planned on having more Q&A time, and I'm going to answer questions until they actually kick me out of here. So, questions? Yes?
Are you familiar with the movie Memento?
No, I'm not.
I wondered if you thought it was accurate.
Oh, Memento.
Memento, yeah.
Yes, I am familiar with it.
About a guy who can't read memory trying to solve a murder mystery.
Yes, yes, and he has to keep writing notes to himself.
Yeah, what did you think of the portrayal of his condition?
Memento is one of the ones that's considered by neuroscientists to be pretty accurate.
I like the movie.
Yeah, yeah. Even Limitless, which seems a little far-fetched, is actually thought well of by neuroscientists because it's a real drug. Yeah.
50 First Dates.
And yet, and the same thing on the rom-com side. By the way, I was describing a Hallmark movie when I was talking about the illusions. But yeah, no, it's a valid thing because what they based it on is H.M. Henry Molaison wrote himself notes. That's how he did what he did, is write himself notes. Yeah.
I'm an expectant parent. What are the best ways to encourage brain growth in babies and kids?
One of the things we do, and it actually runs all the gamut from animal research to human studies and treatment, is environmental enrichment. It is the case of use it or lose it. There are biological reasons why things happen, but in terms of developing the best, read to the kids, have music playing. It doesn't have to be classical music. Have music playing. Be in a family that talks. I am always fascinated to have a conversation with my sons. I've got a son who failed high school senior history, his high school history, world history. He took the AP test and maxed because his failure was they didn't talk about it enough interactively in the class, but we sit and start talking about some battle. We were talking about Battle of Jutland, and Brian starts going in on, well, you know, the such and such happened at the Battle of Denmark Straits and so on like that. And I go, how do you know that? You didn't do that well in history. He says, well, I studied it on my own. Encourage that. Encourage looking up. As far as TV and other entertainments, they're okay, but the downfall is the 5-minute attention span. Sesame Street and everything else for kids caters to a 5-minute attention span, which is great when they're really young, and as they get older, they need to be concentrating longer and longer and longer. My youngest son would not read anything but chapter books, 20 pages, lots of pictures, and everything like that, until Harry Potter. And he did not want to wait for his mom to read it to him, and so he jumped from I'll only read 20-page books to I've just read 350 pages in the last 3 days. And so encourage that. That's what you want. Other questions? Yes?
On the other side of that gentleman's question, what about keeping your brain active when you get to, I just turned 70.
Play games. Mahjong, cards, Sudoku, Wordle. My father was 91 in the hospital. The day he passed, he was in the hospital. He and I were talking about the road systems, the highways around New York City. I pulled open Google Maps and was confirming that he, in fact, remembered everything perfectly well because he stayed active, always doing something. He did puzzles. He read books. He liked historical type fiction that he would, and autobiographies and the like. And he always encouraged me to do it. And so these are things that don't require you to get out and move. However, mobility is wonderful, and mobility helps your brain. And it doesn't have to be going out for a jog, taking a ballroom dance class, or anything that gets you moving. Go out for a walk, take a Tai Chi class, or any of these type things. They're wonderful. They'll help a lot.
I'm wondering what your thoughts are. I'm sure everybody's heard of these new class of medications for type 2 diabetics that work on the GLP-1 hormone to regulate insulin. It corrects insulin resistance mainly. But in practice, they also started to find that it has an effect in the brain on the hunger centers, giving a feeling of satiety and muting hunger signals, and weight loss has just been crazy.
One of the most fascinating things of the past 10, 15 years is the gut-brain axis. We're learning that what happens in the gut has a lot to do with what's in the brain. There are nerves that run down, but there are signaling chemicals that come back. GLP-1 works almost entirely in the gut, except for now we're finding that the feedback through several of those hormones that are involved in hunger and satiety are now manipulated by GLP-1. So the GLP-1 is actually not having a direct effect. It's having an indirect effect by modulating the gut-brain axis, which is a fascinating thing. And there's more to come, because it turns out that the bacteria that we have in our gut also affects that axis as well. And that is something that is hot new topic developing.
Yes.
And
gut dysbiosis has been targeted as one of the underlying causes for chronic depression.
Right. On the other hand, GLP-1s cause diarrhea, they cause nausea, and they have to be titrated. I had some very good luck. I'm type 2 diabetic. I had some very good luck with my GLP-1s, and then they tried the higher dose, which would have helped me lose weight, and I can't tolerate it. And my sister can't tolerate it at all. I couldn't tolerate the higher dose, so we backed it off and went to something else. But I will tell you that simply getting my blood sugar under control has done a lot for my ability to focus and think and things like that. And I am watching my time, because I don't want them to kick me off the stage, but I'll keep going till they do.
My firm belief in that is that diarrhea is what helps you lose the weight.
It actually is what gets rid of the sugar. The diarrhea is because your gut is now full of glucose that wasn't there before, and then it ferments.
Yep.
Yeah. And there was a question right behind you, and then I'll come back to you, Rich.
Yeah, so
Probiotics help.
I mean, the old memory, I know in the last, I think, year or so, they've been saying some of the early studies with Alzheimer's has been kind of maybe based on some wrong assumptions. Is there new directions in that, or?
Yeah, so that's,
with 2 minutes you have.
so in 2 minutes, the major assumption, the major working theory for the past 5 years at least has been that Alzheimer's disease is a buildup of beta amyloid, an abnormal protein. It's protein that's normally there, but there's an abnormal variant, and it builds up and it can't be cleared out. And all of the drugs that have been developed, even the antibodies that have been developed and marketed in the past 5 to 10 years, have been targeting the beta amyloid hypothesis, and it's simply not working. There is shifting attention to tau protein, which is another abnormal protein, has a different accumulation pattern, different cells and everything like that. And it may be that we were fooled with too close a target, but it was a wrong target. My theory is, don't mess with the drugs, because you don't give a drug to an area of the brain. You give a drug to the entire body. And it's going to have side effects. It's going to be metabolized by the body as you move forward. We're working on ways to go right at the part of the brain. What's the technology going to be? Is it going to be an implant? Is it going to be a chip? Is it going to be a thingy in the brainy? Or is it going to be some non-invasive technology? I'll come to you and then we'll close out.
Okay. Going back to the prosthesis, Lissa and I were prepping for your panel on our drive in.
You would. I'm not surprised.
And we were talking about the difference between conscious decisions moving, reflex motion, and muscle memory. So kind of what are the differences, how do they process differently, and how does the prosthetic take that into account?
Complex decision is here using pathways that have different information every single time. Once you actually develop a routine, that's programmed into the parts of the brain that you're constantly pulling back a fixed memory and applying it each time. Muscle memory is a bit of a misnomer because it has absolutely nothing to do with the muscles and has everything to do with the memory centers of the brain and the cerebellum. Although that's trope number 6, it's not stored in the cerebellum. It's just that cerebellum is involved in creating that memory. And so they are definitely different. They're different types of memory. I'm actually proposing some research that will understand those pathways better. All right, to close it off and get off the stage, one thing. My shipment of books came in slightly damaged. I have 3 damaged books that all they need is to be put under something heavy to flatten them out. They're free to give away. Y'all are welcome. Anyone can come up and take one of the damaged books. And I would be happy for someone to grab a book. And thank you all. This has been fun. And at the same time,